Okay, hi everyone. JP here. Today we're going to discuss different units. We're going to move on to sugars and nucleic acids. And like the peptides and proteins section, it's going to be divided up into three main parts and further subdivided. So today we're going to be talking about, sort of an introductory lecture on the structure of DNA and looking at the properties of some of the nitrogenous bases. All right, so here's the outline for this unit. So as I said, structure of DNA, properties of nitrogenous bases. Then we'll focus later on the prebiotic synthesis of nitrogenous bases and the properties, reactivities, and the chemistry of sugars. All right, so let's talk about DNA. All right, so there's a couple different ways to think about DNA. And I want to give you a couple different perspectives. So I'm going to actually come back to this slide in a little bit. So I'll just put this up here. And what I want to show you is what is known as a biochemist view of the DNA double helix. So you've certainly seen pictures like this before in probably biology or biochemistry textbooks. Here's your double-stranded DNA, and you've learned that it's characterized by a major groove and a minor groove. And you've also probably seen a cartoon representation like this showing that you have A sort of binding to T in a specific fashion and G, hydrogen bonding to C in a different fashion. So you can get, what is known as base pairing specificity. And then you have these five-membered, sugars, and they are linked together by phosphates. So if you look at this view of this DNA double helix, it really captures what I would call a bird's eye view. You're really looking at the DNA from afar and looking at sort of global structural, the global structural elements, okay? And you're not necessarily looking at individual atoms. You're more looking at it from a holistic point of view. So I would characterize a biochemist view of DNA as being, a bird's eye view, and that's a very great way to look at DNA. But as and as you're learning, not just for this course but other things and different things happening in the world today, it's always important to get different perspective, right? So in contrast, if you ask a chemist to look at DNA, they're going to really probably zoom in and look at the atom by atom structure, the connectivity. They're going to look at things like functional groups and try to understand reactivity. They're going to look at things like bond energies and aromaticity and so on and so forth. So if you look at a structure like this from a chemist's point of view, there are some very, very interesting features here. So I'm just going to sort of pick some here. This is our phosphate group, okay? So it's not just the letter P like it was for the biochemist point of view. And what's important about this phosphate ester bond is that it is an overall negative charge functional group on the molecule, okay? So the negative charge is placed between the different nucleosides. So your overall charge on DNA is going to be very, very negative, okay? Polyanionic structure. If we, move away from this phosphate ester bond, you have here a couple of protons, and anyone that studies organic chemistry and understands the intrinsic properties of proton NMR spectra, understand that these would actually come under diastereotopic protons, and these could be visualized on an MR as diastereotopic protons. Okay, so moving on, if we kind of look at this molecule here, or this entity here, we have, this five-membered ring here, and then attached to this sugar, we have what is called an N-glycosidic bond, okay? And so you have a carbon-nitrogen bond, and let's call this our N-glycosidic bond. And N-glycosidic bonds are usually relatively stable, but if we look at some of the properties of sugars, and as we'll learn later in this unit, there is some movement from the oxygen donating some electron density here, thus perhaps allowing for the removal of this N-glycosidic bond. Okay, so what I mean by this is that this is not a fixed bond forever. There's actually perhaps some lability here in which the oxygen can come form an oxonium ion and you can get removal of that carbon-nitrogen bond. So if that were to happen, then you could generate something like here, right, which is called an oxocarbenium ion, which is really just a fancy way of saying a carbocation that is stabilized through resonance by an adjacent oxygen. Okay, and as we'll learn later, oxocarbenium ions are very electrophilic. They are the center point reactivity uses for not just sugars, but other types of derivatives as well. This also has, a resonance structure where you actually have the carbocation on this carbon here. Okay, so moving on, let's maybe talk a little bit about the heterocyclic base here. This one here is called, thymidine. But what I want you to see is that we actually have this network of p electrons, p orbitals, and you have this continuous circuit of electrons here, giving rise to the fact that these heterocyclic bases are actually aromatic in nature. So they're very stable structures. And lastly, the thing I want to just highlight here is that, and this will become more important when we look at the chemical and biological synthesis of DNA and RNA. In order for these to come together and react, we need a nucleophile to react with an electrophile. So at this position here, since DNA is comprised of phosphodester bonds via a 3'5' linkage, the 3' hydroxyl here is actually a pretty good nucleophile, especially for enzymatic synthesis, and therefore the electrophile here would be your phosphorus. Okay, so that's how this would work. So that's your nucleophile, that's your electrophile, and that's how you could get these two to react together. Okay, so we'll definitely touch base on these as time goes on, but as you can see, the chemist's perspective is really quite different. Atom by atom structure, functional groups. So some things chemists think about is, can we control the reactivity of some of these molecules? Can we synthesize them? Can we understand them? And that could be done by chemistry. Can we also perhaps redesign or make oligonucleotides? And could we even further develop oligonucleotides that have chemical modifications? So similar to the peptide unit in which we looked at peptomimetics and we understood the chemistry, can we apply that to redesign oligonucleotides? Okay, so, and you could think of other really interesting questions as well. All right, so let's talk a little bit about bases. All right, so not bases from a band, but bases that are part of heterocyclic chemistry. And the one that I want to talk to you first about is a base called pyridine. So pyridine, you may have used this in the lab. One of the things with pyridine is that it's actually has, it really smells really bad. It has a super high, well, not super high boiling point, but if you do heat it up or even spill a little bit or don't use this in the fume hood, it does have a pretty bad smell. But what makes it really useful is that it is an aromatic compound, so it's very stable, and it also has a great ability to pick up protons. So it's a very useful reagent for organic chemistry. Okay, so pyridine, let's draw in the p orbitals. And you can see each of these pi bonds here, well, I gotta move my hand out of the way, are sharing 2 electrons, okay? So if you actually count the number of electrons, pi bond is sharing 2 electrons, you actually have 6 electrons, and this is actually aromatic. The definition of aromaticity, if you recall from Organic 1, follows the 4n plus 2 rule. So if n is equal to 1, systems with 6 electrons that are in a continuous circuit like this means it's aromatic. If n is equal to 2, then 10 electrons, 14 electrons, 18 electrons, etc. So this pattern of number of electrons in a continuous shared sort of circuit make these very stable. Now, let's take a look at pyrrole. Pyrrole looks similar to pyridine, except it's missing a carbon. So let's look at the p electrons, and or the p orbitals, and put the electrons. So you can see you have 2 pi bonds here, each sharing an electron. And the nitrogen here, okay, actually has an electron here too. Okay, it has a couple of electrons. So in fact, pyrrole has a continuous circuit of 1, 2, 3, 4, 5, 6 electrons. So this is also aromatic. Okay, except the electrons are on the nitrogen. Now you might be saying, well, wait a minute, what about the nitrogen electrons on pyridine, right? I didn't really talk about that. So the electrons on pyridine are actually not part of the aromatic system. So they're projected out and away, so they're not stacked upon each other. They're projected 90 degrees away and are not part of the aromatic circle, whereas these ones from pyrrole are. Okay, so if pyridine were around, so this is arrow group nitrogen, so this is pyridine, and if pyridine comes and picks up a proton, one of the questions I have for you is, well, what is the pK of the conjugate acid? Okay, is it going to be fairly acidic, or is it going to be not so acidic? Okay, so let's take a look, and let's compare actually pyridine to pyrrole. All right, so if pyridine comes, and as I mentioned, picks up a proton, and becomes protonated, what is the pKa? And similarly, if pyrrole does the same thing, and picks up a proton, what is the pKa? Well, you might say, well, they look kind of similar. They probably have similar pKa's. Now, a question I'm going to ask you is, of these conjugate acids that I've drawn, which one is aromatic? Okay, so here you have 1, 2, 3, 4, 5, 6 p electrons. So this is aromatic. This the nitrogen electrons that pick up the proton is projected out. So definitely there's no contribution from that bond whatsoever. So this is aromatic. And if you look at pyrrole, when it picked up the proton, you actually no longer have p electrons here that can contribute to aromaticity. So in fact, by picking up the proton, this compound loses aromaticity because now you have an sp3 hybridized nitrogen. It's not aromatic. So if you have something that's aromatic versus something that's not aromatic, remember aromaticity is a good thing. Okay. Oh, here's a pyrrole. I forgot those 2 bonds there. So this was aromatic. When I picked up a proton, it lost aromaticity. So in terms of it donating a proton, which one is going to be more willing to donate a proton? Well, the one that's not aromatic, if it loses or donates its proton, it's going to go back to being aromatic. And as I mentioned before, aromatic compounds are very stable. So the pK of this one is actually fairly acidic. It's around 0. Whereas the pK for pyridine, protonate pyridine, to go back to just pyridine, is around 5 to 6. Okay, and remember, this is a logarithmic scale, right? So it's really a difference of 10 to the power 5 difference in acidity. So that is quite significant. This is important. Why are we talking about pyridine and pyrrole? Because as we'll learn soon, the heterocyclic bases like thymidine, cytidine, guanidine, adenine, all of those bases are aromatic, and we need to think about the consequences if we protonate it. Do we lose aromaticity? Because that could influence its reactivity and its function with respect to DNA or RNA. Okay, the other thing about pyridine is that it's a great hydrogen bond acceptor. So organic chemists like using pyridine as solvents because not only do we have a good base, I can accept a proton and also donate it through a conjugate acid. It's great for solubilizing polar compounds. So you have the nitrogen electron here, which is a hydrogen bond acceptor, and it can actually hydrogen bond very well with things like water or other polar molecules. Whereas if you had something like benzene as a solvent, benzene, right, just looks like this. It's entirely hydrocarbon. Benzene with water will not mix, right? So pyridine with polar compounds mixes quite nicely. All right, so in contrast, if we look at some of the nucleic acid bases, so cytosine looks like this. These are called pyrimidines. Thymine, that's in DNA. Uracil is missing the methyl group, and but it's very similar otherwise to thymine. That's predominantly in RNA. These pyrimidines are very similar to pyridine. Okay, I know it's hard to say. These pyrimidines are very similar to pyridine, but yeah, they're very similar. They're aromatic compounds and they're quite stable. So when you actually form these N-glycosidic bonds, you don't lose aromaticity when you protonate it, which is good because you don't want to make something very unstable through protonation and make it more reactive. These compounds here are called purines, adenine, you've seen that before, and guanine. These are also aromatic compounds, and if you look at the the aromaticity rule and draw on your p orbitals, you'll see that these have 10 p electrons, which makes them also quite stable. And these are linked to sugar through these nitrogen stars that I'm showing here. So I'm just showing you the heterocyclic bases. Now you've learned that GC, right, guanine and cytosine, those form 3 hydrogen bonds, right? So we could even draw 3 hydrogen bonds here. So you've learned that, that's ingrained in your mind. AT forms 2. Okay, 2 hydrogen bonds. These interactions are specific, but why are they specific? Okay, so 3 hydrogen bonds, 2 hydrogen bonds. So these are specific because based on their hydrogen bond donor and acceptor orientation, they form a perfect hydrogen bond fit. Okay, so it's really quite remarkable. And what is the evidence, right? So, we've kind of told you that in biochemistry classes, biology classes, but from a chemical point of view, the evidence, there's many different ways you could actually try to understand how this works. If you think of this from an enthalpic point of view, or free energy point of view, if you're taking 2 molecules and bringing them together to hydrogen bond together, that's going to be a very unfavorable interaction from an entropic point of view, right? Entropy always wants to increase. But the payoff is that if you do this and bring these 2 molecules together, For G and C, you can see you have 3 hydrogen bonds coming together. The evidence for this is that if you actually do some type of NMR experiment, you can observe changes in proton chemical shifts in NMR. So if some of you have probably taken Professor Zankina's spectroscopy class. And basically, if you were to take these compounds and put some polar groups on them and use something like, D2O as a solvent, deuterated water, what will happen is that you'll get deuterium exchange with many of these other hydrogens here. So everything will be saturated in deuterium, and deuterium, as you know, will not show up on the proton NMR spectra. Okay, so however, if there's a hydrogen bond here, these hydrogens here, they're kind of locked and stuck between the acceptor and donor, right? So you have, these hydrogen bonds that are really kind of glued in, or they're going to be much slower at exchanging with deuterium. So you can actually see these on the NMR timescale, okay? So these will not exchange with deuterium when doing NMR experiments, whereas the other ones will, so you won't see them. So that's the evidence for some specificity. You can look at short DNAs and oligomers as well, and you can use things like D2O experiments to monitor the strength and how those hydrogen bonds are actually forming. Why does this form? Well, again, this is molecular recognition. That's the way that nature decided to do this. And the other cool thing about NMR is that not only can it be used to characterize your compounds, to look at hydrogen bonds, but you can actually do a titration curve, and you can actually measure an association constant between G and C, for example, or if you had a short oligomer with its complement strand, for example. And you can get an equilibrium constant, and you learn all these great equations in thermodynamics. So delta G is equal to minus RT ln K, which is also equal to the change in enthalpy minus T times delta S, change in entropy. So as I mentioned, entropy of course decreases because you're bringing these two molecules together, but the real payoff is that you get that benefit from those hydrogen bonds. So it's a favorable reaction because delta H for complex formation is 3 times hydrogen bond energy, and as I mentioned, delta S is essentially unfavorable. So if you think about it, if you have a really long strand of DNA and its complement strand, and if these two are to come together, and you have many different hydrogen bonds, that's a really, really strong interaction. There's a huge enthalpic advantage to that. Whereas the entropic penalty is not great, but it's not so bad. You're taking, 2 sort of big molecules, having them come together. But if you have a large strand, the payoff could be hundreds and hundreds and hundreds, thousands of hydrogen bond energy units. Okay, so that just gives you a little perspective in terms of why these things form and the energy, the free energy behind that. Okay, so that's your introduction to DNA. Hope you enjoyed the lecture, and see you next time.